And having done so, it is my great pleasure to introduce Jeff Carstens, who is the curator at the North Central Regional Plant Introduction Station, Ames, Iowa, for woody crop plants. He received his BS in horticulture and MS in entomology. So he has a broad range of training. He started at the NC 7 station in 2003 and became curator in 2011. I am going to turn it over to Jeff Carstens to give us his presentation. Thank you, Jeff.
You're welcome. So thanks, Kim, for the invitation to talk about gene banking activities. Basically, my presentation today is going to cover briefly, some of the germplasm that is curated at our repository, with a focus, of course, on horticulture collections and some of the horticulture project activities. But I want to talk about, some of the things that have changed over the last couple of years. I hope that in covering some of these changes that I've implemented with germplasm management, it might spur some thoughts for potential considerations and or implementations at other MPGS repositories, with the main goal of trying to increase efficiency. So if I was to mention, some major challenges that I have to deal with, it would have to deal with low quantity or quality of original seeds. So as things come in the door, more often than not, we're not getting enough quantity or good enough quality of original seeds. Dealing with woody plants, majority of my crops have very lengthy regeneration cycles. And then it's seeds. If you look at a lot of the crops that have been around for a long period of time, we have a good idea of how long those seeds will last in storage, how to handle them, and et cetera, and specifically what storage conditions they prefer. If you basically look at just about any of the taxa that I deal with, we don't know those answers. How long will those seeds last in the fridge? How long will they last in the freezer? How should we be handling those? Those are all questions that I have to try to figure out, and with the extreme number of different taxa that I have in my collection. Just some historical perspectives. The MPGS kind of dates back to 1898 with that Seed and Plant Introduction Bureau. But our station in Ames opened its doors in 1948. And then, of course, in 1974, it was the reorganization to the National Plant Germplasm System, which encompasses all of the stations that we have here. What's really interesting is you can read our 1949 annual report, which actually states that there were a couple thousand samples that were received within the first year. Our station has about 150 acres located in the southwest edge of Ames. We're considered a Zone 5B, which is categorized as a -20 to -15 Fahrenheit, which is a reminder that refers to the average minimum low temperature over 30 years. And this season we did see -17 degrees Fahrenheit actual temp. And in fact, for one whole week, it did not get above 0 degrees Fahrenheit. And now 3 weeks later, we're sitting here at 60, 65 degrees, and it's supposed to hit 72 tomorrow. We normally see about 36 inches of rain per year, but last year we got about half of that. And just in the short duration of time that I've worked in Ames, we've seen droughts, we've seen floods, we've seen things bloom 4 weeks ahead of schedule than what one might call normal. So needless to say, the weather here is very unpredictable, and it's very wide ranging from year to year. If you take a look at the crops that are in our station here, You'll notice that maize makes up close to 50% of what we have in our holdings. And rightfully so, because we're located in the Corn Belt of the United States. Pointing out that most of our crops here are more agricultural type crops. But I mentioned that, as most of you know, there's a lot of interest in wild crop relatives. So maybe down the road, a large percentage of our holdings will actually have more non-agricultural crops. If you look at a breakdown of what we have in the horticulture collection, you'll notice it's a very extremely diverse mix of taxa. And again, if you look off to the side on the blue portion of the pie there, it just is labeled as other. And that's almost half of my collection that I am curating along with my colleague Andrew Sherwood, who's the new technician that just started working with us a couple months ago. And 15 years ago, if you would have looked at this collection, specifically if you looked at ash, it would have only represented about 3% of our holdings. But because of EAB that was introduced in the US in 2002, that initiated a reactive response to basically start collecting seeds throughout the United States before those genetics were lost to this devastating insect. So if you looked at, say, elms that were wiped out by Dutch elm disease, or chestnuts that were wiped out due to the chestnut blight, you know, those events happened at a period of time when gene banking or seed banking really wasn't happening. But now we do, and we've obtained lots of ash seeds. And hopefully we'll be able to use these genetics for restoration projects, also for breeding projects, et cetera, as we go into the future to try to figure out how to deal with the emerald ash borer. As a side note, you know, thinking about seed banking efforts, you know, as you look at what people are doing and collecting across the globe, a large portion of that is a response to these insect and diseases or habitat losses. And a lot of people are focused on the rare or the endangered species. But, you know, I just want to mention, I really think a lot of us need to be considering about collecting some of the more abundant and common taxa, just because they're easy to find, they're easy to collect. Because once they do become rare or are under threat, it's when they become quickly difficult to acquire. The main concern I have looking at this pie is the fact that our fraxinus holdings have gone from 3% to almost 20% within a 10-year time frame. And so what that means is that, you know, at some point in time, maybe that's 50 or maybe it's 75 years from now, someone's going to have to regenerate those accessions. So not knowing how long lived these seeds are in storage is kind of scary. And, you know, I don't know, we really don't have a realistic plan in the future for how we're going to be able to handle those regenerations. In the horticulture collection, I have both clonal accessions, which are maintained as plants in the field, but also seeds in storage. If you took a look at the collections in total, 90% of what I have is represented as seeds in storage. And the other 10%, as you see down the bottom, this is an example of our willow field. Our willows are maintained as clones, and also the elms. Why? Largely because we've always treated these as recalcitrant species, which means we can't store the seeds. And it's kind of fun to show this picture of the willows because a couple of years ago, someone asked me about an accession that we had and said, what can you tell me about the growth habit on this particular accession? And unfortunately, I didn't have an answer. And I was like, well, we're always cutting these back and maintaining them as stock plants so that we have cutting materials available. And so that kind of spurred a decision to let these grow so we could figure out what their growth habits are. And it's really neat to see, even though we have some species that are the same taxa, or some accessions that are the same species, their growth habits are very different. I don't know if you'll notice in the picture here, there's a couple accessions that look kind of brown. Those are what you would call our Japanese beetle susceptibles. There's two accessions here, and then there's one off into the far back. So thankfully with the folks at Fort Collins, they've been very helpful in backing up our collections through cryopreservation as dormant cuttings. If I had a choice, working with a new taxa or new species, if I have a choice to say I'll maintain it as a clone or maintain it as seeds, I would definitely try to maintain it as seeds. All that time weeding, pruning, propagation, spraying, and mowing around plants in the field takes a lot of time. So only in cases where there might be a specific plant or a group of plants that has very unique traits would mean I would try to maintain it as a clone in the field. So trying to describe growth habits isn't always very easy. And so when we had these clonal plants that reached 10 years in age, I had a student, Lyndall Morley, that actually took pictures at night. And then she took the images and photoshopped them by drawing in where the branches were in Photoshop and then making that to scale. So if you looked at our willow holdings, you can see the branch habits on a lot of our willow accessions that are maintained as clones. And I started thinking about this the other day when I have a couple apples in my own property at home, and there's a couple varieties that I really like based on their branching habits, and they're easier to prune. So I don't know how many people are interested in perhaps branching habits with apples, but because they're more interested in fruits, but it's something to think about. So as I mentioned, it's a lot of work maintaining these. Here's an example of two plants that are very tall and that haven't been cut back. And then right in front of them, you can see that where they were cut back the year prior, and that's the response and growth. So again, back to this, the point is we need to maintain these so that we have good viable cuttings, but it's also a lot of work. Perhaps instead of maintaining these plants in the field, we could just be maintaining cuttings and cryopreservation and distributing those instead. Here's another look of some larger trees that had to get cut down after the 10-year period so that we can start to get more cuttings that are accessible for distributions. Taking a look at our institution as a whole, we have currently about 54,000 accessions, which represents almost 10% of what's in the entire NPGS. If you look at a 10-year average, we're generally acquiring about 450 new accessions every year, and we're regenerating about 1,000 every year and backing up about 750. So while we can continue to maintain these sort of outputs, trying to deal with the growing backlog of accessions that are gradually increasing in number because of the new acquisitions, but also addressing those that are declining in viability over time is extremely challenging. Looking at our distributions, just within the last 10 years, they've doubled. And so, this is an added, not necessarily an added work for me as a curator, but it's added work for the folks that are filling those orders. And as a side note, our staff dedicated to doing this for filling orders has not changed. So they're required to be more efficient as well. So how do they get more efficient with seed distributions? Those of you might know Lisa Burke, who's our seed storage manager, implemented what's called pre-packs. These pre-packs are essentially a tiny packet of seeds that contains the standard distribution amount for that accession. So it can be quickly pulled and shipped rather than trying to count out or weighing a distribution when it's needed. Looking at the horticulture project, the number of accessions that I've acquired since I've been here, it's basically doubled. So when I first started working here in 2003 to where we're at now in 2020, like I said, it's doubled. We've also gone from about 150 different genera that I'm curating to almost 215 different genera. Again, back to the comment about the number of people that are helping curate this. Despite the doubling of accessions since I've been here, it's still just myself as one curator and one technician. So trying to prioritize these accessions when it comes down to figuring out which ones are most critical for saving will be questions that we're going to have to answer as management becomes crucial. While I was preparing this talk, one thing that kind of came to my mind was how do I figure out how to give an example of better management to other NPGS sites? And as I started digging around in some numbers, I started looking at a couple things, and that was looking at the percent active and the percent available for accessions that were received in the 1980s, those in the 1990s, and so on. And you'll see right away that the materials that we have from the 80s, only about half of them are active, and the same for the ones that we received in the 90s, only half of them are active. But why the huge jump for the ones in the 2000 and 2010s? While I tried to get some better numbers to really point out these examples, I found out it was becoming very hard to query these in GRIN. What I can tell you the difference is that a large portion of these accessions that I've gotten in through our doors since 2003 really traces back to the quantity and the quality of original seed that first comes in the door. Historically speaking, a lot of the samples that we got in the 80s and the 90s were probably in the ballpark figure of 50 seeds or less. Or it was for accessions where it was a seed sample represented by a single mother tree. And anytime we get materials that come in as a single mother tree or 50 seeds or less, it's almost near impossible to try to regenerate it for those reasons. So to give you a little background on one project that I was involved with, and this was with Andrew Schmitz. He's the gentleman on the right. I'm on the left. He's with the Brent Arboretum in Dallas Center, Iowa. And in 2008, we started working on a project to sample Kentucky coffee tree across its native range. This species was selected due to its adaptability to urban conditions, drought, and the fact that it had no serious insect or disease problems, which made it a very likely good candidate to replace ash trees affected by EAB, and also oaks that were being affected by oak wilt. Coinciding with this native distribution, Kentucky coffee tree really only comprises a small percentage of what's in the urban landscape, and also the forest canopies in nature, which basically helped justify creating an ex situ germplasm collection for this species. After a few years of working with it, we soon realized that potentially this species may become extinct in nature, perhaps in the next 100 to 200 years. And that's because the prehistoric mammals, the gomphivores, the mastodons that used to roam North America would eat the fruits, scarify the seeds, and disperse them out into landscape. Those animals are no longer around, and so it's very possible that you could see a loss of this species into nature. And since we started this project in 2008, we visited 16 different states and traveled many miles. And currently we have about 85 different populations as represented as seeds in our gene bank. So as I mentioned, trying to get good quality seeds in large quantities is dependent on a couple of things. And one thing that I've learned as a collector is that every species has a very specific time as when it likes to disperse its seeds. As an example, Phlox pilosa, tiny prairie plant, basically has a harvest window of about 5 days. So if you're not out there harvesting and during that 5-day period, you're not going to get any seeds. As opposed to Kentucky coffee tree, which really has a 6-month harvest window from about October 1st through April 1st. But, even though you could start sampling in October 1st, you're likely not going to get any pods off the tree. The peduncle that holds that fruit is so tough, you basically have to cut it to try to get it off. But if you wait into the very last part of winter or even into early spring, these pods like to drop like flies. They are easy to harvest. And so timing is extremely critical. The other thing I'm showing here is a picture of me using what's called a big shot. And this is basically a slingshot where we take a weighted bag tied to a string, launch it over a branch, and then you can start shaking that branch, and the fruits will just start dropping readily. So this is a technique that I've really been using a lot for ash, for Kentucky coffee tree, and just last week we were out harvesting white pines throughout certain native stands in Iowa. So putting timing along with technique, we went with the Kentucky Coffee Tree Project from averaging about 100 to 150 seeds per tree to well over 3,000 seeds per tree. And so, again, a lot of those collections that came in later are active, they've been backed up, and they're available. So not only is timing important of when to collect the seeds, but it's also knowing when to look for that species. The example here I'm pointing out is yellowwood, or Cladastris kentuckia. And this is a picture that was taken down in the Ozarks in southern Missouri. So in 2019, a team of myself and another colleague, we went and looked for Kentucky yellowwood, and we hit it when it was in flower. Why? Well, when it's in flower, they stick out like sore thumbs, and you can see them from almost a quarter mile away. When they're not in bloom, this species is almost near impossible to find. And as you can see in this example, there's only one specimen. There's no way we'd be able to find this any other time of the year unless it's in flower. So we're going out there and marking things when they're in flower so that we can return to them when it's time to collect the seeds and find them by using the GPS coordinates. Before heading out to collect, it's always important to do some homework and make sure that you have numerous specimens to sample at a target site. In this example here at Kentucky Coffee Tree in Oklahoma, we sampled 10 trees, and this was a 1.5 mile stretch. So this line is 1.5 miles. So the more geographic distance I can put in between my samples would ensure that they're genetically unrelated. So not only does this result in more seeds due to the sampling of multiple specimens, which can quickly result in making them available, getting them backed up, but when it comes time for regeneration, we can use a small set of seeds from each of these mother trees and put them together to cross them, because a lot of the taxa that I have are extremely self-incompatible, and if they recognize pollen of similar parentage, the seed set is very low and very minimal. So what does this look like on the inventory table in Grin? I think a lot of people are interested in this to try to figure out, okay, because I could assign 10 accession numbers to each of these trees. But back to this image here where these are all sampled within the same watershed, they all have very similar adaptative traits for the area where they've been adapted to. So in this example, they're all assigned the same accession number, but they're different inventories. And this is a bottom section of the inventory table. I separated this out so I could put it on one page. You'll see here I have latitude and longitudes. These are actually the coordinates for where each of these specimens were collected. So if someone was trying to do some ecogeographic studies and they needed to know the distances between the samples, we can provide that information. It's always fun to look at this too, because you can see here in this example, I'm only making this inventory, specimen number 3, available, simply because it has the most seeds and perhaps the highest quality of seeds. If people do want to request all of these inventories within this accession, I can definitely make those available upon request.
Oh, you keeping the baby so warm?
Yeah, it's so warm.
Yes. If I could have you mute your mic, please, that'd be appreciated. So what are some other ways that I'm trying to increase efficiency at my station? One of those is using what's called an offsite location. Simply an offsite location is another site where I have some plantings. And this location is about 10 miles to the northwest of Ames. And here I've planted a number of woody plants that I will allow them to be open pollinated. And they're being open pollinated in an area where that's the only accession I have of that species. And it's generally a very unique species where it wouldn't be crossing with anything else in this area. And everything around this is basically corn and soybeans. So just within a first couple of years of having these at an offsite location, you can see here I had some dwarf birch that were planted in 2017. And 2 years later, we collected close to 12,000 seeds. Same with Rus typhina, planted 2017. We harvested it 2 years later and had close to 33,000 seeds. Both of these would be backed up, they'd be made available. And as far as I'm concerned, we probably won't have to regenerate these for at least another 75 years. Target populations. This is another way that we're increasing the number of seeds that we have when we're doing a regeneration. Historically speaking, we've used 5x5s, 7x7s, and 10x10x20 type of cages. Normally speaking, we'd put about, say, 50 plants in a cage, in say a 5-foot by 5-foot cage. Well, now we're going through the extra effort to plant 100 plants in a cage in, say, a 10-foot by 10-foot plot, and basically doubling that quantity of seeds to go into that jar. For a lot of woody plants that I curate, such as Cornus moss, Viburnum lintagos, pagoda dogwoods are some other examples. Those shrubs can get very large, usually 10 feet tall with a 5-foot spread in just a couple years. So what we figured out is that, you know, in a 10-foot by 10-foot by 20-foot plot, I can only fit about 24 plants in a cage. So we went to these large pollination cages, which are 50-foot by 30-foot by 15-foot. And now I can put close to 100 shrubs in a plot. And so for those taxa that I had mentioned, which normally would take 7 to 8 years to increase, because I only had, say, 24 plants in a cage, having 100 plants in a cage, I can now get an increase within about 3 to 4 years, or about half that time. I'm always jealous of Dave Brenner. He's our curator for amaranth and other miscellaneous taxa. This is a picture of his regenerations in a greenhouse. And these are wind pollinated amaranths. And he's basically working with a 3-foot by 2-foot plot where he's putting in close to 100 plants in a plastic cage, and he's getting close to 100,000 seeds. Back to my example that was taking 3 to 4 years, he's able to do these regenerations in approximately 3 months. So 3 to 4 years isn't that bad. But if you look at some of the other taxa I have, some of them have extremely lengthy regeneration cycles. This is an example of Kentucky coffee tree and Cornelian cherry that took 22 years and 11 years from the time of planting until we finally got a good seed increase. This is just not doable for trying to regenerate these. And so what are some ways to try to extend out the availability of these accessions? Normally speaking, for most of what we have in our holdings, the seed distributions are shipped out at 100 seeds each. And usually our critical amounts, which are set at 1,500. But you'll see here, I've lowered those distribution quantities and the critical amounts. And so instead of shipping out 100 seeds for Kentucky coffee tree, we're only shipping out 10. And most people only need 10, but if they need more, they can sure request it. But this allows us to have a lot more distributions over the years. And a while back, one of my students, I was working with him, and I said, geez, I said, we're never going to get enough seeds off of this. We should just run back to southern Oklahoma and recollect it. And he kind of looked at me and he's like, what? Are you serious? He's like, well, that would take a couple of days. And I was like, well, think about it. Yes, it might take a day or two to drive down there and collect it and come back, but then it's done. And then, you're not taking care of something for years and years and years, plus trying to pollinate it in our plots. So recollecting in nature is definitely a very, very viable option for trying to regenerate something as opposed to a typical increase. The prediction queries, I wanted to talk about that. There are some ways to consider when we might have to regenerate something, and a lot of that has to deal with the longevity of the seed, which unfortunately I don't know for a lot of my crops, or it might have to do with how often something is requested. So there are some models that a person could use to try to predict when something might need to be regenerated for long-term wise, predicting, say, 10, 20, 30, 50 years down the road. Looking into the future, I think one of the biggest impacts on the management of accessions at any NPGS repository really might be thinking about DNA sampling. Just within the past 2 years, I've noticed that the number of requests coming in folks are asking for, well, can you send me leaf tissue? I really don't want to deal with seeds. And especially for some of the taxa that I have that have very complex dormancies, it might be 6 months before they get germination. So folks want leaf tissue because it's immediately available. They can analyze it upon receipt. And honestly, I can't say I've looked at other projects across the NPGS. I don't know how many other folks are doing DNA tissue sampling. I actually had a discussion this morning with my technician. He was saying something to the lines of, well, I don't think clonals would be doing a lot of tissue sampling because they can just walk out to their field and collect fresh samples whenever they like. I was like, well, that's not quite the point, because if you did a sampling for tissue and you collected enough tissue, say for 100 distributions, you could take those leaf tissue samples and simply store them just like you would seeds or cuttings. And so that's what we're really going to try to do over the next couple of years, I think, is focusing on DNA storage. And the main thing is that instead of shipping out seeds, we're shipping out the leaf tissue. That's saving on our inventories. And so I really think that's something that folks should really start to consider. And the other thing I mentioned, and some changes are coming to GRIN where we can make both cuttings available, DNA tissues available, and seeds available for the very same or single accession on GRIN. So people that are requesting materials are going to have options as to what they really want to request. So mentioning about storage, I wanted to show an example of these are birch accessions where when we received these, and these are 20-year-old samples, when we received these about 20 years ago, we had enough seeds and we decided to split the samples where we put some in the freezer and we put some in the fridge. And I'm really glad we did this because now we're able to recognize that, hey, those samples that are in the fridge are no longer viable. So, this resulted in a huge shift in how we're storing birch to putting everything in the freezer. And so when I see stuff like this and I think about all the different taxa that I have, it's a little scary to say, dang, why are we storing things in the fridge when we should be putting them in the freezer? Part of the issue with putting everything in the freezer, although it may last longer or extend its longevity, is the fact that somebody, if they request a distribution, is that somebody's going to have to go in the freezer. And so we're actually in the phase of trying to figure out, can we put a subset of samples in the fridge for a short period of time, and if they go out, they can be accessed and shipped out. And then if we need to recycle them out, say every 2 or 3 years, we could. But that's a huge management decision to make. Handling, okay. Here's an example of Salix amygdaloides, willows, and Salix serissima. And this is an example of just the pure handling of the seeds. And so what I'm showing you here is the viability of Salix, these two different taxa, where we kept the seeds at room temperature. And you can see with Salix amygdaloides, within 3 weeks of sitting at room temperature, these things are dead. Whereas with Salix serissima, those seeds are pretty good to about 6 weeks, but shortly after that, they're declining rapidly. So, now we know that we really can't wait a day or two with Salix amygdaloides. It literally has to be put in the freezer immediately upon processing. Now let's take a look at an example of Durca palustris, where we're looking at both storage and handling. So this is a taxa where that was collected a couple years ago, and we did some TZ tests, which was the before storage. And then after they were stored in either the fridge or the freezer, or a sample was allowed to after ripen for about 8 weeks and then put in the freezer. Then we did a TZ about a week or two after exposed conditions of storage. And then we did another TZ test about a year later. And now we're looking at 3 years later. And you can see here that the fridge samples are maintaining their viability quite well. So the old saying that colder is always better doesn't always apply. So here's an example of where both handling perhaps and storage is making a difference in viability. So, I really have to take a look at these for all these taxa that really haven't been worked with as far as seed banks, historically speaking. So this essentially concludes my presentation. I hope that you guys that are thinking about strategies as to how to be more efficient can think about how to better target new collections. Making sure that either if you're doing the collecting yourself, that you're getting enough materials and good quality materials, or that you're communicating clearly with the people that are going to be doing the collecting for you to make sure you get that number of seeds or cuttings and that they're good quality. Perhaps some of you want to think about doing some offsite locations so that you can regenerate more than what's just at your site, especially if they're wind pollinated species, and doing hand pollinations is extremely labor intensive. Again, the population sizes is obvious. Thinking about lowering the distribution quantities. I know a lot of folks that are requesters may not be happy about only getting 10 seeds, but if they understand that we're really strained and that we're growing with the number of accessions that we have to take care of, while the number of people that we have to take care of those isn't following the same trend, things have to change. Again, DNA distributions, I really think this is a way to go. If people can have access to these, I think they'll take them. which again doesn't take away from the seed distributions. And again, storage and handling, this is something that I'm always going to have to be looking at, especially when I have so many a diverse group of taxa, and especially longevity. I really don't know what the longevity is for majority, if not all, of the taxa that I curate. And as I mentioned, when you have a large number of accessions that come through the door at the same time, means that they're going to have to be regenerated at the same time. So there's a lot of work ahead of us. I guess you could say that's job security, but we really have to be thinking about how to do things efficiently moving forward. So thank you for your attention, and thank you for this opportunity to present. I'll try to open it up to any questions that you folks may have.
Sure. Thank you very much, Jeff, for that great presentation. You spurred some questions in my mind, I know. Our colleague Jill Bushakra had a question she put in the chat way at the beginning. She says, it looks like your seeds are stored at room temperature. Is that correct?
No. The seeds that were stored at room temperature was simply just to give us an idea of how fast we needed to move on storage for willows. Historically speaking, willows were considered recalcitrants, so we never really stored any seeds on those. But recent scientific publications have pointed out that they are orthodox in nature. It's just that they're extremely short-lived if they're left at, say, room temperature or even in the cold, when I'm, cold meaning refrigerator or 4C. So everything that we are storing as seeds is usually stored at 4C, which is the distribution type of samples. But if it's an original sample, or if it's considered our reference sample, which means, a reference sample is defined as something that may have been increased, but there's no longer a original sample that's being used as a replacement of original seeds. So a lot of the originals and references are put in the freezer. Anything that's short-lived, like the willows, like the birch, those are being put in the freezer. So
Jeff, that slide that you showed at the very beginning of that big room, that's a huge refrigerated room?
That is a huge refrigerated room, and I didn't mention it, but everything on the left-hand side is corn. But yes, that's 4C, refrigerator, and that's the room where we put all of our increases. Because it's a lot easier to go into 4C to work and pull those seeds. I kind of mentioned it during my talk, that we really should be storing our seeds colder to extend out the longevity of that sample. So if I know that storing birch in -18C is going to be good for, say, 100 years, that's where I'm going to store my seeds. Because if storing them in 4C is only lasting about 10 years, then that means I'm going to have to be regenerating everything about 5 years if I'm going to keep them in 4C.
So when you do your inventories of your collection, do you do it at the temperature in which they're being held?
We basically, in the location 1 in the grand inventory table, it will tell you freezer or not. And if it says freezer, obviously it's freezer, but if it says jar, or if it says pre-pack, or if it says end of collection, those are all in 4C. So that's the only way we have to mark those as to what temperature they're being stored at.
But when you actually go to inventory, so like we inventory our seed lots, they're stored in freezers, I guess they're -20 freezers. And when we pull the box out, we have to inventory it, but that's being done at room temperature. And then it goes back in the freezer. But that's not what you do. You actually inventory it in the room that it's being stored?
No. Throughout, and here's, again, back to the handling, we don't really track day to day or hour to hour the handling of these collections. And that's kind of my, bringing that to everyone's attention, is that perhaps we need to start doing that. And obviously I've learned already that we do need to be careful about what the timing is. So, with the salix as an example, or any seed lot, when we process them, they're being processed at room temperature. When we're taking the inventory, we're doing that right after we're done processing them, and that's in room temperature. As soon as we're done with that, depending on what species it is, it may go to the fridge, it may go to the freezer. So like I said, if it's a willow, it's going to the freezer immediately after it's been processed and inventoried. Does that answer your question?
Yes,
I think she might mean too, we go back and do a count, I mean, of seeds
review.
that have been in here. So do you guys go back and do a count to make sure that the inventory is accurate in Grin?
Yes, we do. But that basically that sample is being pulled out of storage, counted at room temperature for that short period of time, which may be anywhere from 20 minutes to an hour, I suppose, depending on how many samples are pulled. And then it gets put right back into the storage conditions that it was in.
Okay.
For
Okay.
samples that are not distributed, okay, so I have a lot of taxa that people don't request in a particular year, sometimes a 5-year period. If those samples are not requested, obviously we don't do a count review on those. It's just the ones that have been distributed, and or I'd have to talk with Lisa Burke. We may only do count reviews on ones that are getting close to, say, that 1,500 or fewer seeds as well. So.
And I have a question on your process. It sounds like you have a whole team of people that just do distribution, and you as curator and your technician manage the plants, the regenerations. So how many people are involved in orders?
So yes, I will give a shout out to thanking everybody that works at NCRPAS, because we do have lots of different team members. There's 2 folks that are our entomologists that help supply us our pollinators. We have a plant pathologist that helps us, doing screenings for crops or assessments of diseases that are in the field. We have Stacy Estrada that deals with handling the orders as they come in and alerting us as curators whether or not, asking us if we want to proceed with the distribution. I have Lisa Burke that's in seed storage that actually goes and fills the seeds for the orders that I approve. So there are a lot of people that are involved with helping all of this. Sometimes I have a guy that's helping, do the mowing around the fields and the cultivation. So we have a mechanic that helps take care of the equipment if it breaks. That allows us, as a curator, as a technician, to really do the hands-on plant work or the database work to make sure our collection is in good order. It's a lot of work. It's a lot of work. And, I totally depend on all those people, which I know a lot of the other NPGS sites out there don't have that sort of support staff. So.
Yeah, we're a smaller unit, and that's true. We have a couple DNA points that just came in the chat room from Robert Kruger. Well, let's see, he was agreeing with Jenny. Excellent presentation. So Jeff, pat yourself, take your arm and pat yourself on the back there. Excellent. Riverside distributes DNA samples upon request. Sometimes that is the only way, depending on regulatory issues. And then a second email or chat came in from April Nyberg at our unit. NCGR Corvallis also distributes tissue for DNA extraction. So Oh, and Jenny Smith from Davis, we send DNA and leaf tissue regularly too. I guess I will add, to get DNA to requesters in Europe, for example, we can't just send them leaves. They have to be, say, put in silica gel, and then they can pass regulations. But that puts the onus for the DNA extraction on the requester, because we don't have enough staff to also do that too. So it sounds like you're a step ahead if you can extract DNA in quantity and put it off for distribution in the future. Do you have any comments compared to that and the desiccated leaf approach that we've been using?
All I can say is that I'd really like to get a better understanding for everybody that's out there in the NPGS that's dealing with leaf tissue samples. Simply to know if you are distributing, that's one thing. The other question is, how many of you are actually storing DNA tissue so it serves as an inventory that can be distributed upon request, so you don't have to go out in the field, collect it, process it, package it, and then ship it. That's where I'm going with this, and I'm because here's the thing. I think, especially with clonals, correct me if I'm wrong, but I think there might be certain core collections or certain groups of accessions that are typically requested. And, for those, at least maybe you might want to consider taking mass quantities of leaf tissue and processing it, because it really doesn't take any more time to process a sample if you're trying to sample, say, 10 grams of leaf tissue as opposed to a single distribution of a half a gram. So if you can do 10 grams at once, and once it's done, then you just pre-pack it and store it someplace. Then it's ready to be distributed upon request. And again, you're now providing leaf tissue year round. So though, at least for me in Ames, we have a good portion of our year is winter. So, someone could request a leaf tissue sample in the middle of winter and then have it distributed if we have it available. So again, if people are strapped for time and you have that sample to distribute right away, you're one step ahead of the game.
Let's see, April Nyberg is in our lab, and she's working. I didn't see Nahala on the roster here. April, can you respond about your comment in the chat? But you'll have to unmute.
So this is April. For a long time, we would collect large amounts of material in like 50 mil tubes and then freeze dry it. But we found that after 3 or 4 years that the quality of DNA and after extraction decreased to the point where it wasn't worth sending it.
Really?
So,
and these were freeze dried and put in the freezer, you said?
they're freeze dry. We stored them at room temperature.
Oh, well,
It's stable and it's stable at room temperature in the dark after freeze drying.
Sure. Would storing them in the freezer last longer?
I bet that it would, but we also have, once you start storing stuff in the freezer, you're storing so much stuff in the freezer that you don't have freezer room. So it depends on what your resources are.
Sure. Well, and I would say that, as an example, ash seeds, they're huge. Kentucky coffee trees, they're like the size of milk duds. Those take up lots and lots of space. So if you were to compare, the amount of space that leaf tissue is taking up, I would think you could be able to compact it down fairly small. I don't know. It would be good to know more about the quality of leaf tissue. If that's the case, you know, knowing what would be the difference if it had been stored in the freezer. You know, and is this different from one taxa to another? You know, I know there's a lot of crazy studies out there that have looked at leaf tissue of herbarium specimens that are 100 years old, and they're still getting some useful materials out of that. So
yeah, it depends on what you're trying to get from it. You can, the older ones you can't get.
Sure. Well, I'm not a geneticist, so I'm not gonna, you know, obviously pretend I know everything there is to know about genetics and handling. But I do know that there are a lot of people that are requesting leaf tissues for analyses. And if we can meet that need that way, then I'm going to try to make our clientele happy.
I was going to jump in. Jenny Smith from Davis just sent in a comment. Jenny, do you want to unmute and just mention about your desiccated leaves and fresh leaves that you distribute?
Yeah, so at Davis, the bulk of what we send is mostly dormant cuttings, although we will send other forms as well, ranging sometimes from seed to fruit. But the other big thing that we distribute is leaves, particularly fresh leaves, because our domestic requesters tend to want the freshest, recently expanding leaves for their work. But sometimes you'll have a requester that can't have fresh leaves for whatever reason, whether it's a phytosanitary reason or it's going overseas. And so then we have to send them desiccated leaves because we just don't have the staff there was a time, I'd say maybe 12 years ago, that we had a bigger genetics lab and we could extract DNA and send vials of DNA. But our staff is just so small now that we don't have the people to be able to do that. But and like I said, the bulk of what we send is really dormant cuttings. Occasionally we'll send some green cuttings and other things, but most of those, or pollen, most of those orders need to be directly coordinated with the crop manager because they are a lot more labor intensive to go get, and they're a little dicier to ship because you have to make sure I do all of the phytosanitary work with the county. We don't send our material to APHIS to ship, so we do all of the certs and everything. And even within our state, we have to sometimes have phytosanitary certificates. So we get a little choosy about what exactly the form that we're going to send. The requester really has to have a good reason for why they want a specific form.
So, you know, one comment that you mentioned is that your requesters tend to prefer fresh materials. I'll be honest, you know, the one thing that just baffles me with GRIN orders is that, especially with me, when I have so many different taxa, I got people that are wanting, you know, seeds for forestry projects. I got people that are wanting seeds for fruit breeding projects. I got people that are wanting, you know, materials for biofuels. So I got a whole plethora of people, and I can't say that there's ever been a really good consistency in people requesting materials and where they really understand what we have and how they're going to get it. So what I'm getting at is, you know, the people that are asking for fresh materials, if you communicated with them, would majority of them confirm and verify that yes, they can only use fresh materials as opposed to desiccated?
So for any order request that comes in that isn't your standard dormant cutting, so I've kind of trained our requesters to, if it's not your standard distribution, that they need to put it in the comments, and then I personally reach out to every single one of the orders that I accept and flat out ask, or I'll forward it onto the crop manager for that crop. If it's something that I don't know the answer and can't coordinate with them, I'll ask down to what diameter size of cutting do you need, especially if they want green cuttings. So I'm only able to do that because I reject so many orders. There is a time where I could not do that. We had thousands of orders going out the door, and we were just lucky we could get them out. And then we hit a wall to where it was literally thousands of requests, and we just couldn't keep up with the backyard growers and the homeschoolers. And as much as that's valuable, we just couldn't do it. And then we started rejecting orders. So now the order numbers are so low that I can take the time to reach out to the people and find out what timing do they want?
Yeah.
What size do they want? What do you really need this for? And it makes the quality of the material better. And that's just kind of how we've done it at Davis.
Sure. Well, anytime you're reaching out to them, there's a connection there, and they know that you're trying your best to get them the best quality materials. So, and they're going to get a product that they can use. So kudos to you for doing that. I know that's a lot of work. It's just that sometimes people just aren't, and plus, like, as you know, Jenny, we're going to start transitioning to having multiple inventories available. So, and multiple forms. So the plot thickens a little bit as we move forward. So.
Well, yeah. And actually, in all honesty, like, it's saved me a lot of time, because before I would have requesters submit the same order requests 2, 3, 4 years in a row. And then I'd have to reach out and say, I've sent you the same material 3 years in a row. What happened? Well, we didn't get the right material, or we didn't get the right form. And so I found that it actually saves me a tremendous amount of time to just reach out from the beginning, that I'm not going to have to repeat their order. And you made a really good point that I also want to talk about, which is the changing the distribution quantities. We do that all the time, because as Colonial Collections, we have some trees that are small, and or we'll have a tree that's very popular. We'll have maybe 15, 20 people that want that one tree. We have 2 copies at best of our trees, and we can't get 100 cuttings. So sometimes we have to reduce that down to, you're going to get 2, or you're going to get 1. And that's just the way it's going to be. So we have a lot of the same challenges as you were going through. I was like, oh yeah, we have that challenge. Oh, yep. Even though we're clonal, they're very similar in the challenges, especially with distribution and deciding how are we going to propagate this. I have a walnut collection that's very old. I need to repropagate that, but I need to know, when is it critical? Where am I going to put it? How am I going to do it? And so everything you said really resonated with everything that we're experiencing at Davis.
Yeah, I mean, I look at just what's happened with me in the past 15 years, how my collection's doubled. I can't imagine, it'd be fun to look at where your collections have gone. Have you guys doubled in the last, 10, 15 years? At some point in time, you're going to be like, well, I don't have any more room. I guess I'm going to have to shut my doors as far as accepting new materials. Or you have to start saying, okay, let's run through the collection and pick out who's a winner and who's a loser. And as you know, we're all into this boat of preserving genetic diversity. That's a little scary, because you can't rub that crystal ball to know, like, okay, that accession, it looks like junk right now, and it maybe doesn't have any useful characteristics now, but it might be the key to resistance for something else down the road. It's a tough gig.
We're actually there. We are walnuts. We have more coming in that were collected from the caucus areas by our geneticists that are coming from quarantine. It's growing. The collection just keeps growing and growing and growing, but we don't have any more space. So we've been moving our spacing to tight, like one foot spacing between our trees, keeping them really small, almost like hedges, just so we could get them in the ground. And we don't want to remove anything, because, like you said, there was a time where people were like, why do you have Juglans microcarpa? It's just a weedy small thing.
Hey,
and then we said, no, we're going to keep it. And now it's like, well, guess what? Microcarpa is like the magic, like, yay. It has resistance that we really need. So it is a challenge. And we keep saying, we need more land. We need somewhere to put it, but we don't want to remove anything. What do we do? So it's again, shared challenges.
Well, folks, I'm going to have to jump in because our time is up, and a few people are peeling out here. I do want to mention, let's see, April Nyberg made a comment about downstream DNA processes requiring high quality DNA. And so again, we try to keep touch with our requesters the way Jenny was talking about, and it depends the requester and if we'll go the extra mile where it's really needed. And then Claire mentioned Claire Heinz, she had to leave, but there's some fascinating points that to explore with Jeff's philosophy for maintaining crop wild relatives. So we all are, let's give Jeff a round of applause if we can find that little reaction. There we go. So thank you so much, Jeff.
You're welcome.
And I have more questions left in my hopper, but there's just not quite enough time to do this. But I hope you
Sure.
can come back and we'll try to arrange another week where people can meet again on clonal issues. So
Well,
thank you very much.
you're welcome. And if there's follow-up email questions, just feel free to email me.
Okay, and bye to everybody.